Thank you for joining today's session entitled Beyond D2 Receptor Blockade, From Mechanisms to New Treatments for Psychosis. I'm Oliver Howes and I'm chairing the session together with my co-chair, Professor Anissa Abidagam from Stony Brook University. And we've got a great panel of speakers. We've got Dr. Ragy Girgis from the Columbia University, Professor Eric Chen from the University of Hong Kong. I will also be speaking. And finally, we have Professor Daniel Lodge from UT Health San Antonio. And we'll be covering a range of clinical and preclinical topics to address this issue that we're discussing in the symposium. So I think it should be a very exciting session. Just a little bit of housekeeping beforehand. We understand that the slides may lag or be slow at times. We apologize for that, but please bear with us. Each speaker will present their slides, and then Professor Abidagam will moderate a Q&A session at the end of the symposium. We realize that you may well have other questions that aren't covered after watching each speaker. And if you do, please email the speakers and they will try and respond. They will leave their email addresses at the end of their talks. And finally, sirs have asked me to say, please promote science and your colleagues and institutions and sirs by posting comments and using Facebook and Twitter, and use the hashtags #sirs2020 and #sirs to really help get us all through these difficult times that we're dealing with at the moment. So thank you very much for joining us. And I'm now going to hand over to my co-chair, Professor Anissa Abidagam, to introduce the first speaker.
Yes, thank you, Oliver, for organizing this session. This is going to be a special treat for all the dopamine lovers out there. And God knows that we need to have a distraction these days and focus on science and take our minds away from what's going on in the world. So it's really an extreme pleasure for me to be here in this session. We have an incredible panel, as Oliver has mentioned already. And I just want to say that this is also a great time to think about dopamine and schizophrenia. This panel will go in depth into some of the regulatory and cellular mechanisms that may lead to abnormal dopamine and schizophrenia, but it's also gonna go into additional conditions that may share dopamine dysregulation with schizophrenia. So for example, high-risk states and delusional disorder and genetic conditions. So it's really a very exciting panel. And I want to thank Oliver for putting this together. Our first speaker is going to be Dr. Ragy Girgis from New York State Psychiatric Institute, Columbia University in New York. Ragy, go ahead.
Right. Thank you, Oliver, for putting this together, and also Anissa for putting this together in the introduction. And thank you all for joining this talk. I'm Ragy Girgis. I'm a psychiatrist at Columbia University. I'm the director of our clinical high risk for psychosis program. I'm going to talk to you about a PET study we did with PHNO, examining dopamine transmission in people at clinical high risk for psychosis, or the psychosis prodrome, as we sometimes say. I'm listing my disclosures here. There are many reasons to examine dopamine and psychotic disorders. A lot of people, including Anissa, did a lot of great work in the past several decades, Anissa and Oliver, in schizophrenia and otherwise, identifying that dopamine transmission is increased in striatal regions and then possibly decreased in extrastriatal regions. These sorts of findings have very important neurobiological and therefore treatment implications. More recently, Oliver and others have done a lot of this work in clinical high-risk populations. The goal, again, being to determine the treatment implications of medications or other strategies for intervening or modulating in dopamine pathways early on or otherwise. Some of the work is on this slide. For example, Oliver with others showed that dopamine is abnormal, generally abnormally elevated in parts of the striatum in clinical high-risk individuals, and this may predict conversion to psychosis. It has in some papers, it hasn't in other papers. Dopamine synthesis capacity and stress-induced dopamine release is elevated in immigrants and CHR individuals in striatum. Guillermo Horga, along with Anissa, Cliff Cassidy, and other people, demonstrated using neuromelanin-based MRI that neuromelanin as a proxy measure of dopamine is related to positive symptoms, like positively related to positive symptoms in people at clinical high risk for psychosis and with schizophrenia. And then about 16 years ago, Anissa and colleagues showed that psychostimulant-induced dopamine release is elevated in the striatum of individuals with schizotypal personality disorder compared to control people. Now, schizotypal personality disorder does overlap substantially with clinical high risk. They are not the same thing, but those two groups do overlap pretty substantially. A few investigators have also examined extrastriatal dopamine in psychosis. Oliver and others examined midbrain dopamine in clinical high-risk patients using F-DOPA, which is a measure of dopamine synthesis capacity. They found that using F-DOPA, midbrain dopamine synthesis capacity is elevated in CHR compared to controls. Anissa, Mark Slifstein, and others used FLB457 and the psychostimulate challenge paradigm to show that dopamine release in cortex is actually lower in people with schizophrenia than control subjects. The ligand we used in our study is a ligand called PHNO. It's a D2, D3 receptor ligand. It is dopamine 3 preferring, and it's an agonist ligand. So this ligand has been used in several studies in clinical high-risk individuals led by Ramina Mizrahi in Toronto. In one study, she observes elevated stress-induced dopamine release in several striatal regions in clinical high-risk subjects. In another study, she reported elevated stress-induced dopamine release in all regions of striatum of individuals who are at clinical high risk for psychosis who don't use marijuana compared to those who do use marijuana. So again, in this paradigm, just to make clear, Romina's paradigm was similar to the psychostimulant-induced paradigm in which people would be scanned at baseline and then after a stimulus. In the psychostimulant-induced paradigm, we would use a psychostimulant such as amphetamine or methylphenidate. In Romina's paradigm, she used some sort of stress, some behavioral stress, and then would scan people before and after the stress. Our goal was to use PHNO and the psychostimulant challenge paradigm to examine striatal and extrastriatal dopamine transmission in CHR individuals. We chose PHNO because PHNO allows us to measure both striatal and extrastriatal regions, including the midbrain and the thalamus. So we wanted to examine these regions at the same time using a psychostimulant challenge paradigm. We recruited 14 subjects at clinical high risk for psychosis. All of these subjects met criteria for being clinical high risk based on the SIPS. And in particular, they all met criteria for the attenuated positive symptom syndrome. We recruited 14 matched controls. We scanned everyone for 120 minutes. We gave PHNO as a bolus, which is typical. Everyone received 2 scans, 2 PET scans, one at baseline and then another scan 60 minutes after a 16 milligram oral dose of methylphenidate. We used an ROI analysis for the regions and an SOTM analysis for the modeling. There were no differences in scan parameters or methylphenidate blood levels between patients at clinical high risk for psychosis and healthy control subjects. All values here are given as mean and standard deviation. There are no significant differences in scan parameters and methylphenidate blood levels between patients and control subjects. Here are the main results of the study. These data are delta BPND. That's the percent change in PHNO binding due to methylphenidate administration. And just to orient people, a negative value means that dopamine was released from the methylphenidate or the psychostimulant challenge and has basically displaced or is competing with the PHNO ligand. That's why we get a negative value, because at baseline, without a psychostimulant, the PHNO or the ligand binds to a certain degree. Once methylphenidate, in this case, is given and dopamine is released, it competes with the ligand. And that's why these numbers are negative, because at the second scan, there's less binding. So psychostimulant challenge, in this case, methylphenidate, led to release of dopamine in all regions in all patients. However, there were no differences between the release in between control subjects and patients in any region except for ventral striatum. So there's a substantial difference in dopamine release in ventral striatum, where there was greater dopamine release due to methylphenidate challenge in clinical high-risk subjects compared to control subjects. Because the ventral striatum is relevant to depression disorders, we examined these groups considering their potential depressive symptoms or diagnosis or disorder. There were no differences that didn't change anything. And there were also no relationships between methylphenidate blood levels and delta BPND in either group. We examined relationships between positive and negative symptoms as measured by the SIPS and BPND, and the only relationship we found was that there's a very strong correlation between delta BPND in the ventral striatum and baseline total negative symptoms. So not change in negative symptoms. I'll explain to people or show people what potential changes in symptoms there were in just a minute. But we found a relationship between delta BPND and just baseline total negative symptoms in the clinical high-risk group, in which greater displacement, so greater release of dopamine, was related to less negative symptoms. The next question is, how much did the methylphenidate challenge affect positive symptoms? In schizophrenia, we know that about 50% of patients who receive a challenge with methylphenidate or some psychostimulant experience an increase in positive symptoms. In our case, we examined total positive symptoms before and after methylphenidate challenge. The total positive symptom score before methylphenidate challenge was about 15.5. After methylphenidate challenge, it was virtually identical. We found essentially no difference in positive symptom scores before and after methylphenidate. And we examined them both. We examined them an hour, 3 hours, and 24 hours after methylphenidate challenge. These data are for the 3-hour time period, but the results were no different whether looking at the more acute or the more subacute time points. Our conclusions are that striatal dopaminergic abnormalities are present in clinical high-risk subjects, as Oliver, Ramina, and others have shown. Striatal abnormalities, dopaminergic abnormalities, precede extrastriatal abnormalities. We are concluding that because in this study, we found striatal abnormalities in the ventral striatum, but we didn't see any abnormalities, for example, in the midbrain in this study. We suggest that the earliest subcortical dopaminergic abnormalities may begin in limbic areas, such as the ventral striatum. We conclude this because our study observed abnormalities in limbic regions. Other studies, such as Oliver's studies, Ramina's studies, have found abnormalities in primarily associative striatal areas, which are the same regions that Anissa and others have found to be abnormal in schizophrenia. This would also be consistent with the population that we examined, which I'm going to get into in the next slide. Taken together with other literature, these findings may reflect that striatal dopaminergic alterations progress in a temporospatial pattern, not just actually striatal dopaminergic abnormalities, but striatal and extrastriatal dopaminergic abnormalities may progress in a temporospatial pattern. They begin in the ventral striatum, limbic striatum, and then they progress to associative striatum and then extrastriatal regions. A few considerations, one that I alluded to before in terms of population differences, we use an exclusively APSS population. In other studies, populations that included BIPS subjects were used. People who have BIPS are considered to be or tend to be closer to the onset of schizophrenia. Our population, or our sample of clinical high-risk subjects, were completely non-substance using, they had negative urine toxicologies, and were naive of any psychiatric medication. Free and naive, actually, of psychiatric medications. This could be related to, the differences between this study and other studies could be related to cellular mechanisms. Obviously, we use the psychostimulant challenge as opposed to a stress challenge or an examination of dopamine synthesis capacity. And of course, especially compared to the F-DOPA studies, we use a different tracer, a D3 preferring tracer that binds to receptors as opposed to a tracer that examines dopamine synthesis capacity. I would like to also acknowledge my co-authors and mentors, Dr. Abhi Dargam, who really showed me how to do this study and worked through the study with me at all times. Mark Slifstein, of course, who helped us with all the modeling and other components of the study. Dr. Lieberman, who provided great support for the study, and a number of other people, Anna, Jody, Larry, and Gary, without whom I couldn't have done the study. We'd also like to acknowledge the support of Doris Duke and the Brain and Behavior Research Foundation, and of course, Columbia University. Thank you very much. And I'm now going to turn the talk over to Oliver.
Okay, thank you, Ragy. Just a point of clarification. I think when Ragy says release, what he means is availability, since we know that methylphenidate blocks the transporter, and this is how it increases dopamine levels in the synapse. So now we move on to the next talk. Thank you, Ragy, for keeping us on time. It is my extreme pleasure to introduce Eric Chen, who is joining us from Hong Kong. He is a professor at the University of Hong Kong. Eric?
Good, okay. Thank you so much for enabling me to join this online presentation. So I'm going to describe a study of dopamine synthesis capacity in delusional disorders. So it's a study that's been carried out by a group of psychiatrists in collaboration with radiologists and nuclear medicine physicians in the Sanatorium Hospital, private hospital in Hong Kong, which has the facility to do PET-MR. So the background question that we are interested in asking is the extent of dopamine abnormality in psychotic disorder, the spectrum of disorder that from schizophrenia to delusional disorder, and even in other psychotic disorders. But we'll be focusing on delusional disorders in this presentation. The question that is being asked is whether these disorders do share common mechanisms like dopamine dysregulation. And there has been controversy arguing to what extent these psychotic disorders are similar to schizophrenia or different from schizophrenia based on psychopathology and clinical cause and treatment response. And so it is extremely important that we also approach the question by looking at one of the most important mechanisms that we can actually get hold of in looking at schizophrenia, that of dopamine dysregulation revealed in molecular imaging studies. So that's the background of the study. And we also are interested by the clinical observations that antipsychotic medications appears to be equally effective in treating delusional disorders as well as schizophrenia. So suggesting already that there are shared abnormality in the dopamine system. And we recruited first episode patients with psychotic disorders presenting to the public hospital services in Hong Kong. So we do have a early psychosis program in Hong Kong, in which people with suspected psychotic disorders are referred for assessment. So we recruited from these services, and the age range is between 18 and 65 years. We do require the subject to be Cantonese speaking because of some of the neurocognitive tests that we have used, but we won't be presenting those results today. We would try to scan the patients within 1 month of antipsychotic treatment initiation. So one of the unique feature of this study is that we are actually doing the dopamine imaging in a pragmatic sort of setting, where we actually collaborate with a clinical facility in a private hospital who actually has been doing dopamine imaging for clinical purposes. So that also is important, I think, because it may lead us to begin to think about actual clinical potential application of these technologies. But it also at the same time constrains the availability of the scanners and so on. So we have to be a bit more flexible and allow for a short time of antipsychotic treatment before the scanning. So the diagnosis was made according to DSM-5 criteria of delusional disorder, schizophrenia spectrum disorder. And we also include patients with schizophreniform disorder, other psychotic disorders in another group of other psychotic disorders for comparison. So, so those are the patients that we are recruiting into the study. We use a F-DOPA PET MR scanning and measure the dopamine synthesis capacity, the KOCC 30 to 60 value. And so those are the details of the scanner we used. And we use actually a region of interest approach to identify the target area in the striatum. So we look at the caudate and the putamen and measure the F-DOPA activities in those regions for comparison with the occipital reference area. So those are the subjects that we've been able to recruit. They are, so we are actually deliberately matching the schizophrenia subject with the delusional disorder subjects with age. So for schizophrenia, these are also first episode patients that are experiencing a later than average age of onset. So they're all first episode patients. For delusional disorder, for schizophrenia and other psychotic disorders, schizophreniform disorders and psychosis, NOS. And but we'll be focusing on the comparison between delusional disorder and schizophrenia. So the delusional disorder group is slightly older than the schizophrenia group. The schizophrenia group itself is a late onset schizophrenia group. And then we also compare all the 3 clinical groups with a control group, healthy control group of subjects. And the symptom measure shows what we expected, that the positive symptoms were comparable, whereas there is slightly less general and negative symptoms for the delusional disorder group, and in terms of negative symptoms, but a little bit more in terms of mood symptoms. So the results that we observe is that there is actually a difference between all the 3 clinical groups and control groups in terms of the OCC 30 to 60 in the caudate area, and as well as the putamen area. So let me show the graph a little bit more. So the 3 clinical groups are not significantly different from one another between delusional disorder, schizophrenia, and other psychotic disorder, but they are all different from the healthy control group. So summarizing the result for the caudate region, the differences were clear. And for the putamen region, the differences were also marginally significant between the delusional disorder group and the control group. Now, looking at the actual graph, one may wonder whether there might actually be a difference between the delusional disorder and the other 2, schizophrenia and other psychotic disorders. But the numbers that we have are actually too small to enable us to say definitively whether there is a difference between the clinical group. So we'll say that what we can observe so far is we cannot detect a significant difference between delusional disorder, schizophrenia, and other psychotic disorders. So this observation enable us to put forward a suggestion that delusional disorder and schizophrenia and maybe other psychotic disorder share similar dysregulation in dopamine synthesis capacity. So to our knowledge, this is the first study to look at dopamine synthesis in delusional disorder, and it suggests a transdiagnostic role of abnormal dopamine synthesis, perhaps as a common psychopathological brain mechanism underlying psychotic experiences. And it also can nicely account for the similar clinical efficacy of antipsychotics for treating delusional disorders as in schizophrenia. So some limitations in our study is that we use a scanning protocol that is slightly different from those in other previous F-DOPA studies, and we didn't use a pre-medication because we were doing the scanning in a primary clinical facility. And our patients, although they were scanned in the early days of treatment, many of them had some low-dose antipsychotic treatments. So none of them were inpatient. So they were, because they actually need to actually be able to travel to do the assessment and travel to the scanning facilities. And other limitation, we didn't have data on the cigarette use. So with these findings, it suggested that dysregulated dopamine synthesis is observable not only in schizophrenia, but in other psychotic disorders. In particular, we look at delusional disorder, it seems to be elevated as well. And these are in support of a hypothesis that abnormal dopamine synthesis capacity is a transdiagnostic features that underlie a range of psychotic disorders. Whether there are any differences in the extent of dopamine synthesis abnormality between the different psychotic disorders, for example, between delusional disorder and schizophrenia, will require further study, maybe of a larger sample size, to see whether, although there are elevation in both conditions compared to healthy control, whether there are actually differences between them. So thank you very much. So that's the end of my presentation. Okay.
Thank you very much, Dr. Chen, for this nice talk. Now it's my extreme pleasure to introduce the chair of the symposium, Oliver House. Oliver is a professor at the Imperial and King's College in London. Oliver?
Great, thank you very much. And I'm going to talk about the role of dopamine in risk for psychosis and novel approaches to target it. You can see my email address at the bottom. If you do have any questions and want to email me, do feel free to go ahead. And that's a picture of me when I had my hair much shorter than it is now. I've developed quite long hair since lockdown. I've got lockdown locks. I want to begin by obviously talking about my disclosures firstly, and here's a list of them. I've worked with a number of pharmaceutical companies, and some of the work that I'm going to present today has been a collaboration with Synovian, although most of the work that I'm going to present today on all of the clinical study is entirely funded by the Medical Research Council and has nothing to do with Synovian at all, just to make that clear. And I want to start by acknowledging lots of people that have helped with the work, particularly Maria Rogdaki, who you can see in glasses on the left here, and Michelle Coquinou, who you can see on the right, who did a lot of the work for the studies that I'm going to talk about. And what I'm going to tell you about firstly is a clinical study in people at genetic high risk of psychosis. And then I'm going to present some work on a preclinical study that looks at the translational imaging in a model for psychosis. So, as you've heard already, that dopamine has long been thought to be involved in schizophrenia, and the dopamine hypothesis goes back at least to the 1970s. And when this review was published, since then, I think we've developed a much more refined understanding of it and begun to understand how many risk factors for psychosis can act on the dopamine system. And here you see a figure taken from a review summarizing how some of the environmental risk factors on the left, but also genetic risk factors might act on the dopamine system. And I'm going to be focusing particularly on genetic risk factors today. And here is a summary figure taken from another very nice review that summarizes how we can look at the dopamine system using imaging. And it summarizes the various imaging targets that we can use to measure this in vivo in patients. And I'm going to be particularly focusing on this measure here, which is using radio-labeled L-DOPA to measure the dopamine synthesis and storage capacity in dopamine neurons, and it's indexing this conversion of L-DOPA to dopamine by amino acid dopa decarboxylase, and then the storage of that dopamine in vesicles, which you can see there. And this is something that, as you've already heard, is elevated in schizophrenia, and on meta-analysis you see that it's elevated with a large effect size of about 0.7. It's also linked to the symptoms of psychosis, both in schizophrenia and it seems in other psychotic disorders, or at least in bipolar disorder with psychosis, as you can see here in this figure taken from a study that looked at both bipolar psychosis and patients with schizophrenia. So this suggests that it is a marker of dopamine dysregulation that is linked to the development of symptoms. And as you've already heard very nicely from Ragy's talk, there's evidence that it's dysregulated in people at clinical high risk of psychosis as well. But I wanted to look at the genetic factors and genetic risk. And from a study that we did a few years back, we found that dopamine synthesis capacity shows high heritability. This was a twin study, so we were able to look at the component that was specific to heritability in dopamine synthesis capacity and parse out environmental components. And we found about 50% of the dopamine synthesis capacity in healthy people is inherited. So this tells us that there is a strong genetic component to our dopamine synthesis capacity in vivo. But we wanted to look at a genetic risk factor for schizophrenia. We've actually looked at several, but the biggest single one is a deletion of about 4 megabytes on chromosome 22q. And this deletion has been known for many years to increase the risk of schizophrenia. There's about a 40-fold increased risk. And around about 40% of people who carry this deletion will develop schizophreniform disorder, predominantly schizophrenia, by the age of about 26. So this is, if you carry this deletion, you have a very high risk of developing the illness. And you can see here that the deletion is quite a big chunk of DNA, and it includes a number of different genes, which you can see listed here. But in black, you see one of the genes is COMT, which is a gene that's involved in dopamine metabolism. So there are reasons to think that this deletion will impact on the dopamine system. Some of the other genes may also be involved in the regulation of the dopamine system, but a lot of them we don't know their role particularly well yet. Many of them are transcription factors, which could have multiple actions. What's really interesting about this bit of DNA, though, is that a few years ago, about 5 years ago now, it was also discovered that a duplication of this part of the chromosome 22q protects against schizophrenia and reduces the risk of schizophrenia by quite a large proportion, to about by about 75%. Following from that initial publication by Rees, there have been 3 other publications that have investigated this, and there's been some replications, although one of the studies didn't find a protective effect of this duplication. So it's not completely consistent, but still replicated finding and some consistency there that suggests that if you have a reciprocal duplication on the 22q, this protects you from psychosis. So here we have a very nice genetic model, both of risk, but also of protection or resilience. So this made us wonder whether this impacts on the dopamine system. Firstly, we wanted to test whether the 22q deletion would lead to increased striatal dopamine synthesis and storage capacity, and whether this could underlie the psychotic-like symptoms that develop, and whether the reciprocal duplication that's protective might actually be associated with lower dopamine synthesis capacity, and this might mediate the relative protection against psychosis. So this study, which is in submission at the moment and carried out by Maria Rogdaki, recruited 21 carriers of the 22q deletion. And here you see their demographics and clinical criteria summarized. And we were able to recruit 12 carriers of the duplication and 26 controls. And you'll see here that I've summarized the CALMS scores. This is the clinical rating for their at-risk symptoms or at-risk mental state. So a bit like the SIPS and SOPS, this is a measure of subclinical psychotic and schizophrenia-like symptoms. I want to point out that none of the carriers or the controls had ever had a psychotic illness. We excluded any people who'd had a psychotic illness because we wanted to look at people who are at risk or protected or resilient for psychosis, not people who had already experienced a psychosis. And also none of them were on any antipsychotic treatments. So they were all antipsychotic naive when they were scanned. One thing also to note, though, is that in addition to the subclinical psychotic symptoms that they do have, not frank psychosis, but certainly some symptoms there, they also have other psychiatric symptoms, and this is well recognized in 22q deletion carriers, and you can see that they've got some subclinical depressive symptoms there. So what did we find when we looked at dopamine synthesis capacity? So here you see on the vertical axis, the overall striatal dopamine synthesis capacity in the duplication carriers, in the healthy controls in black, and then in the deletion carriers in blue. And you can see that there was a significant elevation in the deletion carriers, both relative to controls and to the duplication carriers. We also showed a significant linear effect of group on dopamine synthesis capacity, so increasing from the duplication through to the deletion carriers. This was in the whole striatum, but as you heard very nicely from Ragy, there's a lot of interest in subdivisions of the striatum. And so here I'm showing you the data from the subdivisions that we've looked at, the associative, sensory motor, and the limbic striatum. The limbic striatum is very similar to the ventral striatum, ventral striatal area that Ragy was talking about. And what you see is that the elevation in the deletion carriers, the people at risk of psychosis, is present across all these striatal subdivisions, which is interesting and contrasts a little bit with what we find in schizophrenia, where we find that the changes seem to be most marked in the associative striatum. Here we're seeing it also quite marked in the limbic striatum, as Ragy told you about, he found in his study with PHNO. We also looked at the relationship between dopamine synthesis capacity and these subclinical symptoms. And here you see there was a positive relationship between dopamine synthesis capacity and higher subclinical positive at-risk symptoms. And you see on this scatter plot, the 22q deletion carriers are shown with the blue triangles and the duplication carriers shown with the red circles. Importantly, there was no relationship between depression or anxiety scores in the deletion and duplication carriers. So this relationship between dopamine and symptoms seems to be specific to these subclinical psychotic symptoms. Finally, Maria also carried out MRS spectroscopy scans to measure brain glutamate levels in the deletion carriers, as this is another neurotransmitter that is implicated in schizophrenia and psychosis. She found no alteration in any of the regions that she looked at in the 22q carriers in their glutamate levels. So this suggests that in terms of the pathophysiology of this genetic high risk for psychosis, it doesn't involve glutamate. So here you see a summary of the findings. Increased striatal dopamine synthesis and storage capacity in the 22q deletion carriers, relatively lower storage, striatal dopamine synthesis and storage capacity in the genetic low risk, the duplication carriers. Sorry for that reminder for me to update my my backups. So here, in terms of this model of what might be going on in psychosis, we've shown evidence here that the 22q deletion, the single largest genetic risk factor for psychosis, is associated with elevated striatal dopamine synthesis capacity prior to the onset of psychosis, in some of these individuals probably preceding it by quite a few years. We've also looked at some other genetic risk factors for schizophrenia, including the DISC, and found associations with much smaller but still significant increases in dopamine synthesis capacity, and you can see those summarized there. So this is an indication that dopamine might be involved in mediating through the genetics of psychosis might act on the dopamine system. But I now want to talk a little bit about how we might target dopamine regulation and dysregulation to prevent psychosis. And to do this, we wanted to develop a model of the dopamine pathophysiology of psychosis, in particular this striatal dopamine synthesis abnormality. This was informed really by, not by Special K the cereal, in case you're having your breakfast, don't worry, but actually by some observations we made on people that use ketamine, which is called Special K, on the street. And we found that regular ketamine users who were then abstinent when we measured them showed ongoing subclinical psychotic symptoms, despite the fact that ketamine had washed out of their system. And there's lots of other evidence that ketamine can induce psychotic-like symptoms. So we developed a model of subchronic ketamine administration. So here you see repeated ketamine administrations given to mice over 5 days. They're then washed out of ketamine, so a bit like those abstinent ketamine users, and then we scanned them to measure their dopamine synthesis capacity. And there's lots of evidence that ketamine, of course, can affect dopamine, but we don't know whether it will affect dopamine synthesis capacity and whether this will persist after it is washed out. So here are the data from this study that Michelle did. And in the first cohort of mice, you see that even after the ketamine is washed out, you get a persistent or elevation in dopamine synthesis capacity measured using the same PET approach that we use in patients. And actually quite a similar effect size to what we see in patients with schizophrenia. So that's very interesting because it tells us that ketamine is reproducing some of the pathophysiology of psychosis. But of course, what we didn't know was whether this was mediated by dopamine neuron activity. So we wanted to test this. We used a chemogenetic or DREAD approach to selectively inhibit midbrain dopamine neurons. So here you see in green staining for TH, a dopamine marker, tyrosine hydroxylase, and then M-cherry. And we used a construct, this humanized muscarinic 4 receptor that was selectively expressed in dopamine neurons and injected into the midbrain so that it is selectively expressed. And here you see that, there's a high percentage of the dopamine neurons were labeled with M-cherry, which was the marker of the expression of this receptor. And we showed very low levels of off-target expression as well. And what's more, this receptor responds to CNO, which is a ligand that otherwise is inactive in the mouse brain, to inhibit those dopamine neurons. And here you see electrophysiological recordings from these dopamine neurons in mice expressing this receptor. And then when they're given CNO, this blocks the firing of the dopamine neurons. So then we wanted to test whether we could block the effect of ketamine using this DREAD model and giving the animals CNO at the same time as ketamine to see if we could block it. And here are the data from that experiment. So you see the control group in black and a CNO control group in blue. And then you see in red when we give ketamine and saline, we reproduce that increase, elevation in dopamine synthesis capacity that I showed you in the first cohort of animals. But then in purple, when you give CNO to block the firing of midbrain dopamine neurons at the same time as giving ketamine, you can see that we're able to block that effect of ketamine on striatal dopamine synthesis capacity. So that shows us that something that modulates dopamine neuron firing could potentially block this effect on striatal dopamine synthesis capacity and even prevent it if we were able to selectively modulate dopamine neuron firing. What could we do what could we use to do this? Well, here's a figure from a review that we wrote looking at various targets, but one that I want to talk about now today is the trace amine receptor 1, which is present in dopamine neurons and regulates their activity. And here you see some data from previous studies that shows that a trace amine 1 receptor agonist, RO, shown here in this figure labeled B, is able to reduce the frequency of dopamine neuron firing. It doesn't occur when you have TAR1 knockout mice. And also if you use TAR1 agonists, you're able to reduce dopamine levels as well. So this is evidence that TAR1 can manipulate dopamine neuron firing. Does it affect striatal dopamine synthesis capacity? Well, in an initial proof of concept, we tested that in our ketamine model. And here are the data from that experiment. So again, you see in black, a control group, in gray, a control group for the TAR1 agonist, which is a compound called CEP856, which was given to us by Synovian, and they worked with us on this study. And then you see again in red the effect of ketamine on dopamine synthesis capacity, that there's an elevation again in another cohort of animals. So this seems very reproducible. But finally in pink, the interesting finding is that giving CEP856, the TAR1 agonist, alongside ketamine, you're able to block the effect of ketamine on dopamine synthesis capacity. So proof of concept that it's possible to modulate the pathophysiology in terms of dopamine system that we're modeling with this ketamine model. We also wanted to look at whether it's possible to modulate this system upstream of dopamine neurons. And there's lots of theories that actually dopamine dysregulation is perhaps a final common pathway to psychosis, but it's alterations in systems in the cortex that lead to this dysregulation, and alterations in the parvalbumin interneurons that regulate cortical pyramidal neurons are thought to be part of this mechanism. So we wanted to see whether we could block ketamine's effects, and we know that ketamine binds to NMDA receptors on these parvalbumin positive interneurons, and this could be part of the action of ketamine as well. And we wanted to see if we could block these effects of ketamine by activating parvalbumin interneurons. So here we use the same chemogenetic approach, the DREAD approach, but this time rather than targeting dopamine neurons, we targeted these parvalbumin neurons using parvalbumin Cree mice and injecting, instead of a receptor that inhibits the activity of these neurons, a receptor that activates these neurons into the ventral subiculum and the prelimbic cortex of these mice. And we showed that CNO activates these parvalbumin interneurons, and here you see recordings from parvalbumin interneurons, and then when we gave CNO, that increased the activity of those interneurons. And then finally, that giving CNO to activate those parvalbumin interneurons at the same time as ketamine is able to reduce the effect of ketamine on striatal dopamine synthesis capacity. So again, this is evidence that it's possible to modulate this final aspect of the pathophysiology in our model by targeting upstream factors that might regulate dopamine firing. So just to conclude, what I've talked to you about today and shown you is evidence that people who carry the 22q11 deletion, single largest genetic risk factor for psychosis, show higher striatal dopamine synthesis and storage capacity, whereas the duplication carriers, who seem to have a lower risk of psychosis, at least in the majority of studies to date, also show lower dopamine synthesis capacity. So this suggests that risk and protective genetic factors have opposite effects on the dopamine system, and again support the idea that dopamine could be the mediating the risk to psychosis and the final common pathway to psychosis. I've also shown you evidence that a ketamine model is able to induce the same elevated dopamine synthesis capacity that we see in patients, measured using the same PET technique. And finally, that it's possible to alleviate this using either a TAR1 agonist or via activating parvalbumin positive neurons in cortical regions. And this provides some proof of concept of potential new treatment and preventative strategies that could in the future be used even to prevent the onset of psychosis in people at risk if this dopamine mechanism is key to the development of the illness. So thank you very much for your attention, and here's my email at the bottom if you have any questions that aren't covered in our Q&A session at the end. Thank you very much.
Thank you, Oliver, for this amazing talk. So much data, always exciting. What we're going to do now is go to our last speaker today, and that is Dr. Daniel Lodge from University of Texas Health, UT Health in San Antonio, Texas. Dan?
Thanks, Anissa. I'd like to start by thanking the Schizophrenia International Research Society for the opportunity to present virtually. And I'd like to thank Oliver for including me in this symposium. And what I'm going to talk about are hippocampal and thalamic pathways that regulate dopamine system function in rodent models, and how we can use this information to develop potentially new therapeutic approaches for psychosis. So these are my disclosures. I have nothing that is relevant to the work that I'm presenting this morning. So as the last presenter of this symposium, the dopamine hypothesis of psychosis really needs no introduction. However, it is one of the oldest hypotheses and suggests that hyperactivity in this system may contribute to positive symptoms. This is based on imaging studies of increased presynaptic dopamine function, the observation that antipsychotics block dopamine D2 receptors, and the finding that psychomotor stimulants can induce psychosis-like symptoms in both subjects with schizophrenia and also in the general population. The one caveat in this hypothesis is there doesn't seem to be overt histopathology in the dopamine system of patients. There are histological alterations, they're just in places that are upstream of the dopamine system. So my overarching hypothesis is that a better understanding of pathophysiology will lead to truly novel therapeutic approaches for the treatment of psychiatric disorders. And to do that, we really need rodent models. And there's a number of different rodent models. We can have acute pharmacological models, and one of those was just discussed by Oliver. You can give psychomotor stimulants, things such as amphetamine, phencyclidine, or ketamine, which are known to produce psychosis-like states in humans, and you can recapitulate that quite readily in animals. There's genetic alterations that can be modeled in animals, particularly things such as 22q11. And then the models that we typically focus on are developmental prenatal manipulation models. We think that schizophrenia is a developmental disorder, and so we want to model that aspect. And the way we do this is by giving a drug known as methyl azoxymethanol acetate. This is administered specifically to pregnant rats on gestational day 17. It produces histological, neurophysiological, and some behavioral alterations that are somewhat consistent with those observed in schizophrenia in humans. But the benefit of rodents is we can really get in there and measure what's happening at a single cell level, and obviously we can manipulate the system to see how that functions. So to measure dopamine neuron activity, we put glass microelectrodes into anesthetized rats, and we can actually target individual dopamine neurons that are located in the ventral tegmental area and the medial aspects of substantia nigra pars compacta. We can identify dopamine neurons based solely on their electrophysiological characteristics. So this is an example up here of an individual dopamine waveform. They have a broad action potential. They often have this initial segment spike that other cells in that region do not. So once we put our electrode in the VTA, we record 3 patterns of activity. One is the firing rate, how fast these cells are firing. The second is burst firing, or the pattern that these cells are firing in. And the third is termed population activity. This is the number of cells that are firing spontaneously. So I stole this slide from Tony Grace. I think it is a nice representation of how we think the VTA and medial SNC is in a quote-unquote normal animal. And that is that about 50% of the cells are not spontaneously active. They're quiescent, they're hyperpolarized from inputs coming from the ventral pallidum. In contrast, the other 50% are spontaneously active, and they're firing at different firing rates and patterns. So to examine this, what we do is we take a glass microelectrode and we move it through the ventral tegmental area of medial SNC in this predetermined grid-like fashion. We make about 6 to 9 tracks. We count the number of cells that are firing spontaneously, we record their firing rate, and we record their pattern. What we have here is a control animal looking at the number of cells firing spontaneously. When we use different models, such as the MAM model, what we can see is that there's a significant increase in the number of cells firing spontaneously. And not shown here is that there is no change in the firing rate and no change in the firing pattern. The only thing that has changed is the number of cells that are spontaneously active. This is not just seen in the MAM model. We've done some work with an inflammatory model. This is prenatal inflammation using poly-IC. It's a viral mimetic. And we've also done it with a pharmacological approach. This is subchronic phencyclidine, and it's consistent with the model that Oliver just spoke about. This is an NMDA receptor antagonist, just like ketamine. So what we see is increases in dopamine neuron population activity, the number of cells firing spontaneously across a really diverse range of rodent models. This is data from a meta-analysis that Oliver published a few years ago now, and it shows quite nicely increases in presynaptic dopamine function across a wide variety of different clinical examinations. And what we were really interested in is how does that correlate with the dopamine neuron activity that we're seeing in our models. And so we don't have access to the scanners that Oliver does, so we used a more rudimentary conventional approach where we modeled what we see with F-dopa uptake with PET-MR by using tritiated dopamine autoradiography. So what we did is we injected tritiated dopa and we looked at background subtracted intensity of that signal in both control animals and in our MAM model. And what you can see in the MAM model is a significant increase in dopamine synthesis capacity. So this mirrors what you just heard with Oliver looking at PET in the ketamine model and in human patients looking at presynaptic dopamine function. This is extremely preliminary data, so that is a big caveat. We're doing more on this to try and work out what that correlation between dopamine activity measured electrophysiologically and dopamine synthesis capacity measured with autoradiography. But what we're really interested in is why. Why is there an increase in dopamine neuron population activity? And my lab, Tony Grace's lab, many other labs have spent a long time looking at the afferent regulation of the dopamine system. We found brain regions that regulate the activity patterns, so peduncular pontine tegmental nucleus, the lateral dorsal tegmental nucleus, all affect the phasic or the burst firing of dopamine cells, whereas other brain regions differentially regulate different activity states. And I'm not going to go through all of this today, but I am going to tell you that the increase in dopamine neuron population activity is directly attributable to increased activity in the ventral regions of the hippocampus. So just to show you some of that data, here we've got recordings from dopamine cells in control animals and in our MAM model. And when we inactivate the ventral hippocampus using tetrodotoxin to turn it off, what we see is that dopamine neuron population activity comes right back down to control levels, suggesting that aberrant input from the ventral hippocampus is actually driving this dopamine hyperfunction. What's interesting about this is it's consistent with the human literature demonstrating evidence for hippocampal hyperactivity in human patients. This was initially suggested using SPECT imaging in the medial temporal lobe, and more recently has been shown using RCBF and RCBB. Furthermore, this increase in hippocampal activity is directly correlated with clinical measures such as delusional severity. So you can see as cerebral blood volume increases in the CA1 area of the hippocampus, so does delusional severity as measured by the PANSS. So now we have a model where aberrant activity in the hippocampus actually drives this dopamine hyperfunction. And can we use this to develop novel therapies? And the obvious way to do this would be to decrease activity in the hippocampus, but we want to do so without altering its normal function. So one way we could do that is to increase GABAergic function. And Oliver just mentioned increasing the activity of those inhibitory parvalbumin cells. Those cells synapse and exert their signal through GABA-A receptors, which are located either in the synapse or extrasynaptically. The extrasynaptic receptors tend to regulate just the tonic activity of the hippocampus, whereas the synaptic are responsible for the actual function of the hippocampus. So our idea is if we could increase extrasynaptic inhibition, we should be able to decrease that hippocampal hyperactivity while maintaining normal hippocampal signaling. The way we've done that is to use a viral approach. We used a viral approach because it gives us specificity. There's two forms of specificity. One, we can target the ventral regions of the hippocampus specifically without affecting any other structures. And two, we can target this using a specific promoter to only those projection neurons, only those glutamatergic neurons projecting out of the hippocampus. The extrasynaptic receptor we overexpressed is the alpha-5 subunit. This is known to be principally extrasynaptic. So what we did is we generated a virus that expresses this alpha-5 GABA A receptor on the CAM kinase promoter. We injected it into the ventral regions of the hippocampus, and this slide just shows confirmation that this approach worked. The GFP is a tag on the virus, so the green cells are those that express the alpha-5 receptor. CAM kinase 2 is a marker for those glutamatergic neurons, and you can see that those are the neurons specifically that were targeted with this viral approach. What we wanted to do is to make sure that this approach worked and that it only affected tonic inhibition. And so what we're looking at here is the change in the baseline. This is patch clamp electrophysiology of hippocampal pyramidal cells. And what you can see is that change in baseline when a GABA A antagonist is washed on is much greater in those that had the alpha-5 GABA A receptor expressed in them. So this is just to confirm that GABA A overexpression does indeed increase tonic inhibition in the ventral hippocampus. So what does it do to hippocampal activity? So this is an electrode in the ventral hippocampus recording the average firing rate of hippocampal cells. The MAM model displays a significant increase in the average firing rate. And when you overexpress an inhibitory receptor, I guess it's not surprising that you get this reversal of that hyperactivity. But what we're really interested in is what happens downstream. So these are the dopamine neurons of the rats that had the alpha-5 put in the hippocampus. Again, here's our significant increase in the number of cells firing spontaneously. And when we overexpress the alpha-5 GABA A receptor in those glutamatergic cells of the ventral hippocampus, you can see this comes right back down to control levels. So here's a schematic on what we think is going on. Under control conditions, you have about 50% of those dopamine cells are spontaneously active. We think this provides a variable gain of function. You can turn up or down the dopamine signal, just like the volume knob on a stereo, based on things such as environmental context. And that changes the salience of that dopamine signal by either amplifying or dampening that signal. In psychosis, what we think is happening is that all of those cells are spontaneously active, which means any stimulus that comes through gets ascribed this high salience. With a conventional antipsychotic drug, these dopamine cells go into depolarization block, and you actually get a reduced dopamine signal. So you still lose some of that normal functioning of the dopamine system. Whereas with expression of the alpha-5 GABA A receptor, we normalize the system, but still maintain that gain. So we hope that this is going to be a better, novel, non-dopaminergic therapeutic approach. And some work we did a few years ago now, again with Tony Grace and Jim Cook, looked at a selective positive allosteric modulator of the alpha-5 GABA A receptor and demonstrated that to have some efficacy in this rodent model. So I focused a lot historically on the hippocampus and its regulation of the dopamine system. The first synapse in this is a projection to nucleus accumbens. And you can see from this schematic that there's a whole bunch of other brain regions that come in and signal through the nucleus accumbens. And that got us thinking that maybe other regions, and the one that I'm going to focus on is the midline thalamic nucleus called the paraventricular nucleus of the thalamus, may also be a novel approach to regulate dopamine neuron activity. So what we have here is a genetic approach or a viral approach where we used a retrograde virus combined with a floxed virus to enable the activation of specific brain pathways. So what we have here is a pathway from the paraventricular nucleus of the thalamus up to the nucleus accumbens. And over here, we have a pathway from the ventral hippocampus up to the nucleus accumbens. And we can modulate these or activate these with systemic ligands. This is dopamine neuron activity. When we activate the hippocampus to nucleus accumbens pathway, we get an increase in dopamine neuron population activity. And this is consistent with what I've shown previously. However, when we activate the paraventricular nucleus of the thalamus, we see essentially the same thing. And when we record activity in the nucleus accumbens while stimulating either the PVT or the hippocampus, we find neurons that respond to both. So you have convergent inputs coming through from the paraventricular nucleus of the thalamus or the hippocampus synapsing on exactly the same neurons in the nucleus accumbens. So that got us thinking that maybe paraventricular nucleus inactivation could reverse this aberrant dopamine neuron activity in rodent models. Here we have the man model. This is our increase in dopamine neuron activity. Here's our prenatal inflammatory model of poly-IC, again, an increase in dopamine neuron activity. When we turn the paraventricular nucleus of the thalamus off, what you can see is that dopamine neuron activity is decreased in both of these models, suggesting that the PVT may be a novel therapeutic target. But we're not going to go in there and turn it off with something like tetrodotoxin. And what we're really interested in is whether we could manipulate the PVT with a pharmacological approach. And in doing some reading, what we realized is the PVT shown here receives a really dense innervation from neurons that contain the neuropeptide orexin. And all of these green terminals are orexin terminals in the PVT. And you can see that there's much more expression around the paraventricular nucleus than there are in adjacent regions of the thalamus. Moreover, previous studies have demonstrated that orexins actually increase the activity of neurons in the paraventricular nucleus of the thalamus. They do this because the orexin receptors are GQ-coupled receptors. They're excitatory receptors. So the idea is that if orexins endogenously increase PVT activity, then giving an antagonist should be able to decrease PVT activity. And there's actually an FDA-approved drug, suvorexin, which is a non-selective orexin receptor antagonist that is used clinically for the treatment of sleep disorders. So our idea is quite simple. Let's give a dual orexin receptor antagonist and see how it affects dopamine neuron activity. Again, here's our man model with a significant increase in the number of dopamine cells firing spontaneously. And following administration of this orexin receptor antagonist, this comes right back down to control levels. So in summary, here's a schematic, an oversimplified schematic, where we have the hippocampus and the PVT providing innervation of the nucleus accumbens, which then regulates dopamine system function. In the MAM and other models that we've looked at, the ventral hippocampus is hyperactive, and that results in hyperactivity in the dopamine system via this polysynaptic pathway. One of the ways we can target this is by reducing the activity of the ventral hippocampus. And I've shown you data that overexpression of the alpha-5 GABA-A receptor decreases tonic inhibition in the ventral hippocampus and reverses that aberrant dopamine system function. Another approach is to target neurons that are projecting to the same region. And the paraventricular nucleus is one of those, where a FDA-approved non-selective orexin receptor antagonist decreases PVT activity and reverses aberrant dopamine system function. So you can see from the schematic that this one reverses what we believe to be the primary deficit by targeting the hippocampus, whereas this targets adjacent systems to have the same net effect. So it gives us multiple independent ways of regulating dopamine system function. So in conclusion, aberrant afferent regulation of the dopamine system may represent a key pathology in schizophrenia, and targeting this pathology may ultimately be an effective treatment for schizophrenia. And I've put in here in parentheses other psychiatric diseases. We've moved into stress-related disorders and other disorders where psychosis is comorbid, and we're starting to see similar effects in those systems. So with that, I'd like to thank the people that actually did the work, Hannah, Stephanie, Jennifer, and Angie. I'd like to thank my collaborators, my financial support. I'd like to thank you for your attention. And if anyone has any questions, please feel free to email me. I'm more than happy to answer any that you may have. So thank you very much.
Thank you. Well, thank you all for some exciting and fabulous new data that just, in a way, raises a multitude of questions in my mind. So I have a very privileged position of being able to ask all my questions, which never happens in real life, right? So why don't I start with, maybe I'll go in order, and then you all could respond to any of the questions. So the first one that I would like to address is the one we started with from Ragy, was the data about the prodrome, the CHR, showing increase in methylphenidate induced change in PHNO binding, which basically reflects the fact that when you block the dopamine transporter, you look at the effect of synaptic dopamine on tracer uptake. And what Ragy sees is that in the mesolimbic system and the ventral striatum, patients that are in the CHR stage have more dopamine availability than controls. So Ragy, I want to ask you, what is the relevance of the studies in this patient group if we don't know that they're going to actually become schizophrenic? So in a way, this in my mind is a very heterogeneous group, and it's possible that these patients will just go on to just be just eccentric people, or even be just disturbed adolescents or young adults that are going to be okay. Do you have any information on their conversion, on whether the people who converted were the ones that were most abnormal? And, you know, Oliver obviously can also comment on this.
Sure. Thank you. This is an important question. I mean, I guess there are several, I'll keep it short, but there are several reasons how it's relevant. First, just in terms of the number of people who've converted so far. 2 have converted, 2 of the 14 have converted so far. A few of them are lost to follow up. There was, in the 2 converters, there was no difference between converters and non-converters. So baseline dopamine, methylphenidate-induced dopamine reuptake inhibition does not, in these 2 compared to 12 patients, differentiate who does and who doesn't convert. But we didn't share the data because we're still waiting for follow-up on some of them, and the sample size is so small. So it's still relevant. I mean, this is actually a big, question in the field about how important it is to focus on conversion to psychosis versus this high-risk state, which is relevant to many psychiatric disorders. Since we still don't know how to treat these people, we need to learn about their neurobiology, again, to lead us in the right direction.
Okay. Yeah. Another very exciting finding in your data was the inverse relationship to negative symptoms. And just, I don't know if you're aware of that, to remind you that we had observed this inverse relationship in our AMPT study in patients with schizophrenia. So this is the second time there is a kind of a finding of an inverse relationship in the ventral striatum between negative symptoms and dopamine. It just occurred to me, like, do we know which of those negative symptoms was most related to dopamine abnormality? Because the thing with the ventral striatum, if you have excess dopamine, it seems to increase the risk for psychosis. But if you have less dopamine, it seems to bring on more negative symptoms. And it's this kind of like bipolar effect of dopamine in the ventral striatum. I think we need to explore this more. It's fascinating that the same dysregulation could have 2 opposite effect, or can contribute to the pathologies of schizophrenia in opposite manner, if you follow what I'm saying. Maybe Oliver also want to comment on that.
Yeah, well, it seems all consistent to me. Definitely this is consistent with your own findings. I think Romina found the opposite in her data, that there was I guess in this case, however you want to say it, but in her case, with stress-induced dopamine release or whatever happens, the opposite. So less negative symptoms with less negative symptoms was related to
more dopamine, more stress.
More dopamine.
Yeah, okay.
In her subjects, and I think that was actually in the cannabis sample.
Yeah.
Either way, yeah, it seems consistent. But what I was going to say was, I actually have not looked at the relationship between dopamine reuptake inhibition and the individual negative symptoms.
Yeah,
So
yeah,
will...
something to explore. Okay, that takes me, I'll come back to Eric, but that just because it's relevant to Oliver. Oliver, should we revisit the mesolimbic hypothesis of schizophrenia?
I think
too soon. But you also had data in this regard. I think in your 22q11 deletion study, which by the way is a masterpiece. I want to congratulate you on this study. It's an amazing accomplishment. You also find quite a bit dysregulation in the ventral striatum.
Yes, I think what you're alluding to is that starting with your work in the early 2000s, you identified that the associative striatum seemed to be the major locus of the dopamine changes in schizophrenia, and that's something that we and others have also found and since then as well. So it seemed like this old hypothesis, this mesolimbic hypothesis of schizophrenia, was being debunked. But then science often surprises us, and Ragy's data certainly says that actually there is something going on in the ventral striatum. Don't forget about me too soon. And as you rightly point out, we also see that the changes in these 22q deletion carriers are present in the limbic striatum, the ventral striatum, to a similar degree as the associative striatum. So I think it tells us that there's still a lot for us to learn about what's going on in dopamine in the development of psychosis and schizophrenia. It seems that in the established disorder in schizophrenia, it is more marked in the associative striatum, but then perhaps in the development of the illness, there is, as Ragy said, the ventral striatum is more involved. What I think perhaps is that you get this progression from ventral to dorsal striatum with the development of the illness, and that actually fits with what we know about the anatomy of the loops between the VTA and substantia nigra and the striatum, which go from the ventral and anterior parts to the more dorsal and caudal parts. And as you heard from Dan, it fits also with some of the animal models as well, which suggests that perhaps initially you've got this overactivity, particularly in the ventral system, perhaps related to stress, but then it begins to spread into the dorsal striatum, and perhaps that becomes hardwired in some way, and which is what we then see pick up in people with established schizophrenia, and why it's most marked in those people. And of course, it's the more dorsal striatum that is involved in habit formation, and perhaps that's why the subclinical psychotic-like symptoms become delusions and become fixed in the minds of our patients. But of course, this is this hypothesis that needs testing, and we really need longitudinal studies to look at this. We were able to rescan a small number of people, at-risk subjects with F-DOPA a few years ago, and that seemed to suggest there was some evidence of further spread into the more dorsal striatum as people develop psychosis. But we need to do this more to really understand the temporospatial pattern of changes.
Mm-hmm.
I could mention this also matches the spread of glutamatergic hyperactivity from anterior to posterior hippocampus. And as we know, the anterior part of the hippocampus, innervates the more, limbic striatum, and the posterior hippocampus tends to innervate the dissociative dorsal striatal areas. So those kind of the topology matches.
I think that's a really good point. Actually, Ragy, if I can ask a question, or is it too early for me to ask?
Make it brief.
Yes. Well, it was a question really, Ragy, about the pharmacology of methylphenidate relative to the stress paradigm and to the amphetamine challenges, and whether that would could give us a clue as to what might be going on in the clinical high risk people. Because of course methylphenidate is blocking the reuptake, but it's not actually releasing dopamine. So it's presumably indexing an aspect of the system. If there is an increase in the clinical high risk, then that would reflect the fact that if there's overactivity specifically in the ventral part of the striatum where you see it, you've not got the overactivity in the more dorsal parts. Perhaps that is, you're looking at something that's a little bit different to the amphetamine-induced challenges, which are actually also releasing dopamine changes. So it's perhaps telling us that that early on there is this overactivity in the ventral striatum. You may already have alterations in the dorsal striatum, but they're not seen by methylphenidate because it doesn't capture that aspect, but you would see them with amphetamine challenges. So I wanted to ask you what you thought about that, and if you had any other comments on the pharmacology of methylphenidate.
Sure. I mean, and Ines and probably others know more about this than I do, but yeah, maybe the dopamine transporters are differentially affected in schizophrenia or in the development of schizophrenia. I think that's what you're saying. So definitely, I forget who the first author was. I was actually by someone else who saw these data earlier, sent some papers showing that methylphenidate might actually release dopamine similar to amphetamine, as opposed to just preventing the reuptake. But I mean, the short answer is yes. I'm not an expert on stress-induced dopamine, whatever it does.
But one question that comes up is, are there differential distributions of the DAT across the subdivisions of the striatum and schizophrenia versus controls? I think there's very few studies of DAT in schizophrenia, and definitely I don't remember studies that have looked at the subdivisions. So this would be an important question to look at. Let's just move on to other topics because we have so many and so much interesting data to discuss. Dr. Chen, your delusional disorder, that is just such a difficult category to recruit and study. So again, I really admire you for this effort. You discussed, I think you mentioned something about the stages of the illness, but I couldn't capture it well. The age group that you presented is in their late 30s, early 40s. So how long have they been sick, and was there any relationship to the duration of psychosis?
Right. So they are all first onset patients, delusional disorders and schizophrenia patients. So we are comparing first onset delusional disorders with a group of relatively late first onset schizophrenia patients. And the duration of untreated psychosis in the two groups are different in that the delusional disorder patients have a longer duration of untreated psychosis. They have been ill for longer before they present for treatment compared to the first onset, late onset schizophrenia patients. So, so I think peculiar to this group is that for both the late onset schizophrenia and delusional disorder patients, their etiological factors behind the illness may be different from the typical sort of earlier onset schizophrenia patients, and there is some we look at the contribution of family history and also of life events on this group, and find that for the later onset patients, there is relatively more contribution of life events compared to family history. So suggesting maybe the environmental factors plays a more important role in the onset of this group of patients.
Okay. Okay, very good. Let's move on to Oliver. So you had a clinical and a preclinical piece to your talk. In terms of the clinical, the 22q11, so you find dysregulation of dopamine, but glutamate, if I understand correctly, you don't see any differences with MRS measures, which is a little bit surprising to me. I think this was MRS in the cortex, right? Or was it in the hippocampus? But either way, considering, you know, that this is a genetic effect and so many functions are abnormal in this disorder, you know, patients have medical issues, neurological issues, and then the psychosis. I would have assumed that there's just more involvement, especially in the type of circuits that we heard about from Dan, regulating dopamine, that you would see an abnormality in glutamate that would be, you know, as striking as what you see with dopamine. So what are your thoughts on that?
I think that's a really good question, and we looked in the anterior cingulate cortex and the striatum and the thalamus in this sample, and we didn't see anything across any of these regions. And I agree with you. We're expecting to see some changes in glutamate. In fact, we didn't see any hint even that there were changes. I think the other point perhaps to make is that in schizophrenia with MRS, it's not so clear cut that you get large changes in schizophrenia in general with MRS. Now, whether that's because of what we're measuring with MRS, because it's of course measuring levels of glutamate in the entire voxel, not just in the synaptic component, and perhaps that's not what we need to measure. We need to measure the release of glutamate, or something to do with it, more specifically to its synaptic involvement, to really pick up what might be driving schizophrenia, and likewise in the 22q deletion carriers.
I wonder if looking at GABA, maybe if GABA was measurable, if MRS tools were, you know, sophisticated enough to allow us to measure GABA, this may be a critical factor, because you know, as Dan has shown, the effect on glutamate may be, I mean, the glutamate dysfunction may basically mediate the pathology by affecting the GABA system. And that's maybe more proximal.
I agree. And actually, of course, what we need is we need to look at the balance between excitatory and inhibitory inputs to really understand what the circuit mechanism might be. So ideally, we'd be measuring GABA as well as glutamate in the individuals, and in several brain regions, to try and understand how they might relate to each other. And it may well be the balance between GABA and glutamate is disrupted. That said, I mean, 22q, of course, it is a very big risk factor for schizophrenia, but I don't think it's modeling probably the pathophysiology that we see in idiopathic schizophrenia, which, you know, I think it could well be quite different in terms of its cortical changes to what we're seeing in 22q, where you have a very defined genetic lesion affecting a number of proteins. And it could well be that in 22q, you get something that's quite targeted on the dopamine system, whereas in the majority of schizophrenia, actually, you're looking at a process that is, as Dan described, more diffuse and cortically mediated, but they have the same underlying common effect on the dopamine system, and hence risk of psychosis. And the other curious thing about the 22q is that there are still some people that don't develop schizophrenia despite having exactly the same genetic lesion. So clearly, there are either compensatory measures going on here, or there are other factors, whether it's the polygenic background in that individual that varies, whether that's, so maybe, you know, 22q carriers also need to be carriers of other risk genes affecting perhaps the GABAergic function, or whether it's environmental factors. We just don't know. We need to do the longitudinal studies to follow up our 22q participants to find out.
Yeah, so you bring up the important point that there is so much heterogeneity to the disorder that there may not be one neurobiological circuitry explanation to all of it. Which takes me to Dan. Dan, first, I want your thoughts on, I mean, we saw two examples in the TAR1 example that Oliver gave, and I think in one of yours where we see that dopamine cell firing corresponds well to dopamine levels. I think it was your MAM model. So the MAM model and the TAR1, there's a good correspondence between dopamine cell firing and kind of F-dopa, dopamine synthesis. But is this a straightforward relationship? Do we know that for sure, that when you have excess dopamine cell firing, that it actually will reflect on dopamine synthesis, on the activity of those enzymes? Because these are like really clear. Well, you know, I wonder if that is a well-accepted fact. You increase dopamine cell firing, and that's gonna drive the enzymes to work more and get more of dopa, or, you know, more dopamine levels, etc. Can you comment on that?
Yeah, it's a really good question. So is it a well-accepted fact? It's not a fact. It's probably the most parsimonious explanation that we have. And so that's exactly why we decided to go about measuring dopamine synthesis capacity, at least the way that we do it, because in an animal, you can modulate those things. So we can modulate different dopamine neuron activity states and look at how that affects synthesis capacity. So the stuff that we've currently got, you know, it's on film, it takes a long time when you're not, you know, in a PET scanner. But we have gone in there and modulated the number of cells. And if we bring that back down to control levels, does that change dopamine synthesis capacity? If we change firing rate and pattern, what does that do? And then what's the temporal relation between the two of them? I don't think this is an acute thing. So, you know, if you drop dopamine activity, I don't think synthesis capacity changes on that millisecond timescale. I think it's something that takes longer. You know, that enzyme activity probably follows that, but takes longer. So it's a really good question. It's the reason we started to look at this. I don't have an answer right now, but I'm hoping to have one in the next 6 months.
Yeah, this is really fascinating. I want to ask you about the focus on the PVT in the thalamus. That is a very interesting new area in a way. I don't think people have been focusing so much on looking at the effects of the thalamic regulation of dopamine cell firing. And you bring up the point of orexin's role. Especially that we know that there are sleep disturbances in schizophrenia, that I think makes it even more relevant to study this, this area. Can you tell us more about where is orexin exerting its effect on what type of cells, and how is that kind of modulating dopamine?
Sure. So, you know, the orexins have more of a widespread distribution throughout the neuraxis. So, you know, I don't want to give the impression that they're only located in the paraventricular nucleus. But when looking at the thalamus, the PVT itself has a dense innervation of nucleus accumbens. So the same regions that the hippocampus is targeting are being targeted by PVT. And those same regions are innervated by the orexins. And we know that when you record the activity of a PVT neuron and you put orexin on it, the activity goes up. And if you block it, the activity goes down. So because we already have FDA-approved compounds that we can use to sort of test these hypotheses, we thought that would be a good first step. There are more selective drugs for the two different receptors. There's an orexin 1 and an orexin 2 receptor. In the drug abuse field, this is getting a lot of attention. And so there's a lot of drug discovery, specifically around the orexin 1 receptor. And not in the MAM model, but in a model that we have of stress-induced dopamine hyperfunction, that orexin 1 seems to be what's reversing the dopamine hyperactivity and associated behavioral alteration. So we've looked at things like pre-pulse inhibition of startle, which you can also normalize by an orexin 1 receptor antagonist.
So a question to all of you. The data you have presented seems to suggest that regulating dopamine firing, decreasing dopamine synthesis, should be enough of an antipsychotic mechanism of action. Are we ready to say that? Is there no need for a D2 blockade? And any one of you can jump in and share your thoughts on this.
while I'm already talking, I think, it's a good enough reason to look at it. We need more translational studies to see if any of these novel hypotheses actually have legs, if these could be game changers in the field. And so is it enough to get rid of the D2s and jump all in on a TAR1? Not yet, but it's certainly enough evidence to start looking at these in clinical trials.
So my own take on this, and then Oliver, is that I think that this may work before we have a dysfunction of the D2 emerging due to antipsychotic treatment, that's for sure. So these therapies would need to be tested in antipsychotic naive patients, because in our own data, we saw that there is a functional abnormality in D2 beyond dopamine levels and beyond dopamine release. And I have a sense that this may happen, as we saw in the meta-analysis, Oliver, that we published together, that the increase in D2 levels was present only in patients who've been treated before, not in the drug-naive patients. But this may also correspond to not just an upregulation in number, but maybe also some supersensitivity to dopamine stimulation. So I would suggest that these therapeutics, this novel type of therapeutics, may work best in patients who have not been exposed to antipsychotics. Although, the synovian data argues that it's possible that it would work in previously treated patients. But that to me is a critical point to pay attention to. Oliver?
Yes, I agree that with you that there is something going on in the D2 and the postsynaptic side of the system as well that we need to think about, and whether it's heterogeneity in some patients or whether it's a iatrogenic, a drug-induced effect, we don't yet know. But I would, just to come back on your point about whether we need to target early or not, actually I think you've got data that suggests that we don't, because of your study with AMPT. That was in patients, it included chronic patients, I believe, and that reduced psychotic symptoms in those individuals.
Yes, but is it enough? Does it reduce it enough? Yeah.
Well, I think
that's more my question.
Does it reduce it enough?
And in most patients, I think there's obviously an effect on psychosis when you lower dopamine availability. But I wonder if that is a mechanism that will be sufficient. Because, for example, there's this new drug, the VMAT inhibitor, valbenazine. So that reduces dopamine stores, reduces dopamine availability, resorpine, and drugs like that. The way to test it would be, would these drugs be sufficient to treat patients' psychosis on their own? And I think it's an important question that I wish somebody would try to address. Oliver?
I agree with you, yes. It'd be great if the funders are listening, if they would support. It would be very useful for the field, but as a real proof of concept to test this, and to test it both in patients early in their illness, but also in patients that have been treated with antipsychotics for many years, where we might have D2 upregulation and other postsynaptic effects, because then we do see this breakthrough psychosis emerge in some patients, and we desperately need new treatments for them. Now, possibly targeting the upstream regulation of dopamine might be a way of helping these patients, because you could reduce the dopamine dysregulation in the VTA and substantia nigra, and allow the D2 dysregulation and upregulation to correct, and that might actually help them with their psychosis and also side effects from long-term antipsychotics.
So I don't want to deprive you from the opportunity of asking questions among each other. I know we don't have a whole lot of time left, but we do have a few minutes. If you have questions for each other.
I've got a question for Dan, if I may. Dan, fantastic data and very fascinating. One thing I wanted to ask you about was, I think both in your GABA alpha 5 overexpression model and in your thalamic data as well, this corrected the population overactivity in the MAM model didn't seem to have much effect on the control animals, which surprised me. I just wondered if you had an explanation for that, and if you think that this system is quite homeostatically tightly controlled, so you only get an effect where you've got a dysregulated system.
Yeah, especially in anesthetized rats. So, you know, the contribution of cortical structures under anesthesia is reduced. And so when you go in there and reduce them further, you really don't see anything. We've got a lot of behavioral data that I didn't go into, and there are certainly times where we manipulate hippocampus or other systems, and we actually see deficits in control animals, but we alleviate deficits in a model. And, you know, that makes sense. If you've got sort of some equilibrium and you're too far to the right, you push that back into the middle, you're going to, you know, be beneficial. But if you're already in the middle, you push it too far to the left, you're going to generate deficits. And so in the electrophysiology in an anesthetized rat, we typically don't see any adverse effects with these compounds. However, when we look at freely behaving animals, looking at cognitive function, social behavior, things like that, we often do see effects.
Okay. Any other questions or comments? Any concluding comments from any one of you, or, you know, take home message you want to reinforce from your data?
Well, I think the very interesting thing across all the talks is that we seem to see this involvement of dopamine in different conditions, in different models. Dan, you very nicely showed this with poly-IC, as well as the MAM model, and also in a PCP model. And I showed some data for ketamine. So the dopamine system does seem to be key also across delusional disorders in the at-risk. So, I mean, I think the question is this a final common mechanism, or across psychosis, or are we missing something else that's in the mix?
Yes, so it's interesting, in the data you show in the psychosis of bipolar, now the delusional disorder, the CHR, the 22q11. It's almost like you cannot have psychosis without excess dopamine. I think this has become something that's much more clear than it used to be when we started this type of studies in the field. And it's almost like it's the fever of the system. This excess dopamine is related to psychosis, whether it's primary or just an end point. You know, ketamine can get you there, abnormal regulation, poly-IC, MAM, you know, the circuitry type of abnormalities that are supposed to regulate the system go wrong, and we have this dysregulation. That's one aspect. It's also possible that just dopamine abnormalities to start can have effects on the overall circuitry in the brain and lead to abnormal circuitry and abnormality in other transmitters, like it's been shown with the D2 overexpressing mouse. So I think that we cannot at this point rule out one versus the other, and everything is still on the table. And this session, if anything, has kind of rebroadened our horizon. For example, rethinking where does it start within the striatum. And I'm not saying that we have data to question, this, but definitely data that requires additional investigations from that point of view. Okay, well, with this, I want to thank all the speakers today. I want to thank the audience that will listen to this. And please, remember to email the speakers directly if you have questions that we didn't bring up today. So thank you all for joining me. Have a good day.